This gets to how we've been thinking about translational medicine in our group. In a lot of ways, this has been a very low-level study of chemistry, which seems even below what people think about in biology. We've been talking about quantum mechanics, which, that's even below what we think about in chemistry typically. And that's, I think, what's actually really interesting about this problem, because we can actually go and trace features of the quantum chemistry of a molecule, trace that up to how it affects chemistry, how it affects biology, and ultimately how it affects things in the clinic. This is multi-scale modeling at its most interesting, I think. And the typical way I think we think about translational science is usually trying to just get things over the fence towards basic science, translating from basic science to bedside. There's been a little more intention about going from insights we see in the clinic to understanding what the mechanisms are. I think that is an important, valid way to think about it, but in our group we've been thinking about it instead of as a quest to just get something over the fence, think about it from a computational point of view of how we can have a lively and rapid conversation back and forth between the two domains, which is something that we have the ability to do as we get better and better clinical data and better basic science data. So how is that playing out in this case? So first of all, aldehydes have been recently proposed as an important toxic metabolite. What I just showed you is actually controversial in the field because the way how we experimentally look for reactive metabolites aren't good at picking up aldehydes. And so it's been recently proposed that they're an important toxic metabolites that's missed. Now on the clinical side, there's several drugs that are hepatotoxic by hypersensitivity-driven ways, but we don't know why. We don't actually know ahead of time what the bioactivation mechanism is. So we can take these two things arising in two separate discourses and ask now, is there a connection between these? And can we ask, are there drugs that are toxic by this mechanism that we can identify computationally? The first step of that is what I already showed you with terbinafine. Now we can actually look to see whether or not we can see other drugs that fit this pattern that we can study deeper. And so we did that. We looked at molecules that are known to produce aldehydes and looked to see if their aldehyde was predicted to be reactive. We can identify actually quite a large number of drugs. We're talking maybe about 100 or so. And we can then filter this to find out the ones that are hepatotoxic and then filter out the ones that are hepatotoxic by mechanisms that we already know about. And from there, we can get several really interesting cases where this is a potential hypothesis that could explain why that drug is toxic. We can take this further now, too. One of the reasons why it's been hard to even establish whether or not a drug is toxic in a clinical context is you can take a drug through 1,000 patients and never see the toxicity arise. It's idiosyncratic often. Well, idiosyncratic is just a fancy way of scientifically saying we have no idea why. There's no pattern behind it. And the question is there a pattern behind it that we haven't seen yet? And of course, then, almost by definition, it means that it's no longer idiosyncratic. But so then in the end, maybe what we're trying to do here is shrink what the idiosyncratic nature is. And you might say, why does it matter if it's only affecting 1 out of 1,000 patients? Well, 1 out of 1,000 patients is enough to get a black box warning. It's enough to get a drug pulled from the market. It's enough to deny the 999 other patients that benefit from that drug access to that patient. It turns out that that's how severe of an issue it is. It turns out also, too, that maybe it's 1 out of 1,000 among adults, but it's a lot higher among children. And if we start giving it to neonates or to 2-year-olds or 3-year-olds to treat something, maybe it has a much higher burden on them. And if we don't understand the mechanism, we have no idea how to even really test for and look for that. So yeah, we really do want to understand it, even if it's 1 out of 1,000 cases, even if it's 1 out of 10,000 cases, we really want to understand it. Because if you're that 1 out of 10,000 case, and it puts you in a situation when you have severe reactions to it, it matters a lot. It doesn't really matter to you how many other people are in that situation. So we do care about that. And as we understand the mechanism, it can give us hypotheses about why it is that some patients are affected and others aren't. Now, one of the things that comes out of understanding this mechanism is that we have a way to detoxify aldehydes in our body in addition to conjugating with them. We have an enzyme, actually a set of enzymes, I'm just going to simplify it a little bit here, called aldehyde oxidases that are really efficient at finding aldehydes and turning them into carboxylic acids. They add another oxygen, makes them safe, they're not reactive. And that's generally why they've been ignored in drug development for such a long time because you just assume if you see an aldehyde, well, the aldehyde oxidase will take care of it. But we also find out is that there's a large number of drugs that are on the market, commonly prescribed to patients, that inhibit that pathway. So one possibility is that part of the pattern that would make these drugs idiosyncratic is that it's not just that you have to be metabolizing and producing that aldehyde. You also have to be inhibiting your detoxification pathway somehow. So we can now use humans as a model system in the EHR to look for cases where there's patients that are prescribed that drug that forms a reactive aldehyde alongside drugs that inhibit the detoxification pathway and see if they have a higher incidence of drug toxicity in those cases. So that's something that we're in the middle of doing in our group right now. And so now we're getting to the point of going all the way from quantum mechanics now to the clinic, which I'm saying is really the exciting thing about this work, just on a conceptual level, and then also how we're able to engage this on a month-to-month basis in our group.